We all need some energy. And what I'm going to talk to you about is an energy source which is long forgotten because it doesn't seem to be very fashionable. It's an attempt to try to use crop wastes as fuels, and that is my favorite crop, rice. So what we started studying a few years ago, just by chance, is the pathway for lignocellulose regulation in rice, and we saw that it was very similar to that was in Arabidopsis. We could link the systems, and if so, it would be very useful also for other systems like switchgrass, etc. So the way we look at it was lignocellulose from plant feedstocks. The important one is cellulose, which is the largest renewable carbon source available right now, already there. We don't have to go very far. It's got 180 billion tons annual production, and 15 to 30% is in primary cell walls, which is actually composed primarily of cellulose, which is digestible in some ways.
Yeah, perfect.
So the primary cell wall essentially consists of hemicellulose, cellulose, microfibrils essentially out there, and the secondary cell wall around the xylem region, quite thickened, etc. So the primary cell wall is what we could find actually more digestible. And if you look at the composition in cell walls among plants, the cellulose, lignin, and hemicellulose. The cellulose is polymer of beta-1,4-glucan and very high number of molecules, 300 to 15,000. Lignins essentially are polymer from alcohols, which are a bit more difficult to digest. So these are the major global polymers. So lignin essentially is a barrier to get cellulose free and to digest it. Lignin is 10 to 25% of plant dry matter. So it's acceptable that cellulose would be a very good substrate to be able to extract sugars out of in multiple ways. But lignin is a barrier to the enzymatic conversion. It prevents cell wall hydrolysis from the enzymes from accessing the polysaccharides there. So an objective of many labs is to reduce lignin in feedstocks to facilitate conversion. But there's been a story saying that if you reduce lignin in quite a few grasses, so lignin mutants are normally very brittle, so they're not very strong. So there's been a suggestion that actually if you reduce lignin, and actually reduce means completely make it very deficient in lignin, the plant is fragile and weak. But what we discovered is we can reduce lignin using certain ways, and you do not lose strength. And that's the basic first part which I'd like to talk about. The second part is understand actually the pathway in which lignin and cellulose are co-regulated. So the case for crop waste is that among all the wastes among crops, corn, barley, wheat, rice is a major crop waste in terms of the wasted crop, which comprises grains also. And in the lignocellulose biomass, is also the largest in terms of the rice straw. So rice straw, after the rice is harvested, it remains in the field, and actually it's quite detrimental to the field. So they either try to burn it or remove it. So rice straw is not well taken essentially in the perspective of a cropping season. So it would be nice to use that rice straw in many ways, and looking at the total lignocellulose biomass from both the wasted crop, which is the grain, plus rice straw, it's got half the lignocellulose crop waste worldwide. That's so much, 731 out of 1549 teragram. It's usually burnt. And so the question is, what can one do with it? And is there industry which could be worked on it? The potential bioethanol production worldwide from crop waste, if you look at it, rice essentially from the crop waste and from lignocellulose biomass comprises a total of, could give you 491 gigaliters of gasoline per year, which could comprise of 32% of the global gasoline consumption. So the dream is there. The question is, what are the methods? So rice crop waste can produce the 221 gigaliters of gasoline, which is 45% of the global bioethanol from crop waste. So what are the ways in which we could extract ethanol well from lignocellulose, in spite of having lignin out there? Can we reduce lignin and therefore have it easily extractable? Looking at the lignin biosynthesis and cell wall biosynthesis pathway, which comprises of lignin, they start with monomers, for example, from phenylammonia lyase, PAL, number of different components, cinnamate, 4-coumarate, caffeate, etc. And all these molecules essentially can be used for the enzyme coumarate-CoA ligase, and finally reach alcohols, which essentially then can be into these three monomers for lignin. It's not easily visible out here. H-lignin, G-guaisyl lignin, and syringyl lignin. Let's see if we can get that. I guess not. Yeah. So these three lignin monomers, they comprise essentially the end products which form lignin. And some of these, like for example, the G-lignin are more useful than the other lignins. So you need to also then get a ratio of the G to the H-lignins and the S-lignins. So the monocot lignin is a mixture of GH and S monolignol units, and G is more resistant to chemical degradation. So we need to have a ratio in which the other lignin monolignol better. Looking at the pathway of regulation of lignin biosynthesis, the transcription regulatory network, that means genes involved in lignin synthesis and also cellulose synthesis. You have the NAC family of transcription factors, which are the main switches acting on the MIBs. And there are a large number of them. They are redundant. So many of them have not been obtained by mutant studies. The MIBs essentially act on formation of lignin biosynthesis and other cell wall biosynthesis, for example, hemicellulose. These are a number of genes, the proteins, which have been studied in Arabidopsis. The NACs are SND1, NST, VND, and then the MIBs 46, 83. The characteristic is that these NACs regulate the MIBs, and the MIBs essentially bind on the AC element on lignin biosynthetic genes. This is the cis element, and that essentially will switch on lignin biosynthesis. So the opportunity here is to try to regulate these MIB factors and either reduce lignin biosynthesis instead of completely abolishing it. Abolishing, for example, would be structurally not so useful. So our story started with the discovery of a Arabidopsis gene called Arabidopsis AT shine. Shine was discovered on the basis that the activation tag mutant was shiny. And it was shiny because the shininess came from an extra cuticle wax layer, and that wax layer essentially made the plant more impermeable to water. It also was drought tolerant. So we were studying this, and we found out that AT shine transformed into rice. Also, first was Arabidopsis, transformed to rice, gave you drought salt resistance. That was interesting enough. Enhanced water use efficiency and slight increase in cuticular wax. But increase in cuticular wax was nothing much to write home about. So it was something more to it. Anyway, we looked at the gene expression profile of these AT shine, Arabidopsis AT shine gene expressors in rice. Okay, so it's a heterologous system. The original paper was published essentially in Plant Cell in 2004, in which we had shown AT shine was regulating wax biosynthesis. We did not know about this until we did some gene expression profiling of that. We found the differentially expressed genes. And then at that time, we characterized also the shine rice lines, as we call them, which increase water use efficiency and biomass. So wild type and shine, and under drought stress, the wild type and shine, this is still quite succulent. The water use efficiency of the shine lines compared to white lines was higher. The total biomass under drought and normal conditions was also higher. The transpiration essentially of shine lines was low. So it was a good enough characteristic. It's not as good as the higher gene which I showed you about. It had a little bit in suggesting that increase in waxes, if there was any, could be a use in rice. But it didn't seem to be actually the waxes which were doing it. It was probably something else. So when we looked at the microarray, 80 shine microarray in rice, what we saw actually was surprisingly that a whole set of lignin biosynthesis genes were repressed, relative messenger RNA minus. And other cell wall biosynthetic genes, for example, cellulose synthase, CSLs, were increased higher. So he said this is pretty odd. We never saw that actually in Arabidopsis. What's it doing out here? So we looked at the phenotype, and we found out that the shine lines compared to the wild type had increased cellulose. Actually, this came later. We did the next experiment, which I show you first. The composition showed that there was increased cellulose, and these shine lines had reduced lignin. These are quite intensive methods to do it. We needed a large number of plants to do all this, but it was quite clear. And in collaboration with others, we showed that the monomers of the shine lines, for example, the wild type and shine, the GS ratio essentially was high. So the G was more than the high S, and therefore it would be useful for digestibility. We published this phenotype essentially in 2011 in a paper called Coordinated Activation of Cellulose and Repression of Lignin, Biosynthesis Pathways in Rice. The original study on 80 shine was published in 2004 in Arabidopsis. So in some ways, these two stories seem different. 80 shine in Arabidopsis was increasing waxes, and shine in rice was increasing cellulose. How are they related? What can we do about it? We also saw that shine and wild type culm, if you looked at the sugar components, the three shine lines, what you see is that there's an increase in a number of these sugars, rhamnose, fucose, not increase, actually only xylans and glucose. So xylans are part of hemicellulose and glucose part of cellulose. So increase in these two in the lines suggested that there was both a change in a polymer of glucose and a polymer of xylan. And glucose and xylose are main sugars of cellulose and hemicellulose. So these, along with the gene expression data, suggested that what we've seen out there was real. There was a change in cellulose, and the phenotypes confirmed them. So we stained the shine lines of rice with fluoroglucanol, the wild type, stain essentially the lignin red, and the shine here do not stain. So there's less lignin here in the cell wall. Also here you see that in the shine lines this all white, whereas the red staining ones all over is much more in the wild type. And conversely, when we stain these stems with cellulose stain, which is chalcoflow, you see that in the shine lines there's much more brilliant fluorescence of the chalcoflow because it stains more cellulose. This is quite a sensitive stain, and you have to do it comparatively. But we confirmed that then with other methods. So ultrastructure of the wild type and shine, you had in SEM, scanning electron micrographs, you see the shine had a thicker cell wall, and even in a transmission electron microscope, you see the shine lines here. This is shift over, have a much thicker cell wall, middle lamella too. So shine had more deposition of cellulose in the cell wall. Now, predicting what would be a possibility of how shine could be regulating these transcription factors involved in secondary cell wall synthesis. We know the NACs are there, the MIBs are there. Where does shine act, if at all? And so we started studying them. We know that the downstream genes, that was the the genes involved in the lignin pathway and the cellulose pathway. Those genes were up and down regulated, in lignin up and lignin down and cellulose up. But where is then the connection between these genes and the enzymatic genes? So we looked at, therefore, the transcription factors which are known from Arabidopsis. They're orthologs in rice. The ortholog of VND from Arabidopsis, then this is, and other MIBs. And we saw that those ones were down-regulated. So these ones are the ones which are involved in lignin biosynthesis. Now, the lignin biosynthesis, as you saw, that the MIBs regulate both lignin and other cell walls through the AC element, and then cause formation of lignin. And the AC element, we looked at the Arabidopsis and the rice. We formed this consensus ourselves from these genes which were regulated. They're very similar to the Arabidopsis consensus of the AC element, and therefore they probably would recognize similar cis elements on those promoters. Now, looking at historically, whenever you had reduction in lignin, you had plants which could break very easily. They were also sometimes called brittle mutants. Therefore, we compared the wild type and the shine for whether they were stable or they were more breakable. So we developed a breaking force assay based on a method shown here, and we saw that the breaking force required for the wild type in the leaf and the shine was identical. I mean, there's no significant difference, neither in the culm. So these shine lines were not more fragile compared to the many mutants which are known both from rice and maize, in which when you reduce the lignin, the plant is weak, it is sensitive to diseases, etc. So the 80 shine lines are same as wild type, and why? Because we suggested then that cellulose, because it's the strongest organic molecule as a density basis, the reduction in lignin is compensated by an increase in cellulose. So looking at a summary of what the results were, NACs regulate MIBs, and these essentially work through an AC element and repress lignin biosynthetic genes, and they could be involved in increasing transcription of other cell wall biosynthetic genes. Now the 80 shine works by, for example, down-regulating MIBs or also through NACs. But we suggested that it's probably working through MIBs because the MIB transcription factors activities were seen. And this could cause a reduction in lignin and an increase in cellulose using such a model. So histochemical analysis, TEM, and breaking force measurements suggested that this is the pathway we are seeing, an increase in cellulose and a decrease in lignin. So then we looked at how to put this all together. We looked at homologs of transient factors known for secondary cell wall biosynthesis, Arabidopsis, and rice, and made a network based on all which was publicly available from rice gene expression data sets, 129 samples, 45 groups of network we call RECON. We get a co-expression network of genes, and from there we extracted a network of lignin cellulose cell wall genes. The co-expression networks just gives you a model in which all genes which have similar expression pattern are linked together in a network. So in the hierarchy of which genes essentially are involved in cell wall biosynthesis, we had among 86 predicted rice cell wall synthesis genes and putative regulatory genes based on orthologs. So these are all then the 86 rice cell wall biosynthesis genes, which include genes involved in synthesis of monolignals, then lignins. Then these ones are involved in, for example, the MIBs and the MIBs, and which are known from Arabidopsis, and some which have been known also from rice. So we try to get a correlation or an association between which one of these and these were linked together. And all that, this matrix was part then of a network which we formed, in which the cellulose and other cell wall biosynthesis genes are shown out here. SESA, the cellulose synthase gene, X-glu and beta xylanase, and I don't know most of these expansins, okay, SESAs. And then the lignin genes involved in cinnamyl alcohol dehydrogenase, 4Cl, and PAL, phenylammonium. And here are the different transition factors which are all linked. For example, some of these here are quite linked to others. And SHINE is in the middle of this all. Now, the SHINE, OS SHINE, is we put it there. Although what we had used in our network was studying the network was the Arabidopsis SHINE in rice. So we used that as a proxy for the Arabidopsis SHINE. Using this network, we predicted which genes might be affected. So we suggested a number of MIB genes like MIB20, 43, MIB58, 63, which are known from Arabidopsis. These might be ones which are, they were changed in regulation, might be acted on directly by Arabidopsis SHINE. So we conducted binding assays in which the AT SHINE protein extract was used with promoter fragments from these genes, the MIB genes here. And in each case, we have AT SHINE extract. This is minus anything, minus SHINE protein, no competitor, then a SHINE extract, and then a competitor, homologous competitor. So example you see out here, when the presence of AT SHINE, you see a bound probe, and there's a gel retardation. So in each case, there's a retarded band suggesting that there is AT SHINE protein is binding to that particular promoter fragment, which we predicted on the basis of these GCC box motif to be present in that fragment. So at least in the in vitro system, it was predictive of the gene regulatory pattern we saw. So in all, SHINE was working by potentially repressing NACs, repressing MIB genes involved in lignin biosynthesis, and activating MIB genes involved in cellulose synthesis. So this was our prediction, and I'll show you how when we went ahead to prove that. So this then the reduction in the MIB genes involved in lignin biosynthesis would reduce the lignin biosynthetic genes and cause the reduction in lignin. Increase in this would increase the cellulose. So we made a number of constructs in rice. We had done both anti-silencing construct and an overexpression construct of rice SHINE in rice. So this we hadn't done before. Earlier story was with Arabidopsis rice. But the silencing construct did not work, but the overexpression construct of SHINE worked. So what we saw then actually surprisingly that it was not overexpressing the SHINE, but we saw that it was repressing essentially the SHINE. So this is just the SHINE under the 35S promoter, and it was repressing then the host rice SHINE gene. That means it was acting as a sense suppression line. So in this sense suppression line, what we see, the OS SHINE sense suppression line, this is wild type, and this line has got much thinner cell walls. So the thinner cell walls, that's where actually you'd expect more cellulose. In the other story with Arabidopsis SHINE, we had thicker cell walls because more cellulose. Here we have thinner cell walls. It's more fragile compared to this thicker area. The rice SHINE, OS SHINE ortholog this was first RNA-seq analysis, showed upregulated processes, downregulated processes in which a number of them seemed logical. Cell wall, macromolecule, cytoskeleton organization, etc. We take out specific genes which we know, genes involved in lignocellulose pathway, and a number of genes which are involved essentially in the lignin pathway, CAD, 4Cl, some of the MIBs, etc., are upregulated in this sense-suppressed OS shine. And the hemicellulose pathway, this involves cellulose synthase-like genes, are downregulated. Trying to see whether the shine, the natural shine, would be involved in regulation of the lignocellulose pathway naturally in the plant, we try to make a correlation in the regulation of the cellulose pathway genes and shine in different accessions of rice. So when we looked at the accessions of rice, we could see the putative expression of OS shine is high in the, well, in the leaf, and in some cases also in the culm. And looking at a particular target, this is cellulose synthase-like gene, we found a high correlation between the regulation of OS shine and cellulose synthase-like in different accessions, suggesting that probably the expression of shine could be involved in the regulation of the cellulose synthase-like genes in rice in general. So then what we ended up doing was looking at the potential targets of rice shine, which were identified from RNA-seq analysis. So the phenotype essentially of the rice shine was that they were very weak plants. There was a change in stomatal conductivity, and it was weaker. So what we saw is that there was a change also in OS shine, and these were found as targets for OS shine. And stomatal conductivity and photosynthesis was something which we had seen also in which when we put the Arabidopsis shine expressor in rice, we could see good water use efficiency and better photosynthesis. So it suggested that in some ways OS shine was working like the Arabidopsis shine, but not in all cases. The negative effects we had seen was in flowering time and cell cycle genes, because the plant was small, the plant flowered early, etc., and the tillering also was changed. So at present we are left with using, the studies what we have out here, and we have shown binding for some of that. I haven't brought that out here because it still is raw right now. It's a graduate student's work, trying to complete it. But we are seeing that some of these are targets of shine naturally, and we still have to then see how well OS shine regulates multiple components of the plant biomass and lignocellulose pathway. Tillering is involved in biomass, flowering time is involved in biomass, including photosynthesis and cellulose. So we expect that actually what shine is doing is more broader regulation essentially of these processes, plus involved in lignocellulose also. So the perspective is essentially of engineering cellulose, which is at present still a possibility. Cellulose is the largest biomass annually, and it could produce 1 billion tons, which is 97 million of biomass, in a vision to replace the 30% of the liquid fuels with ethanol. So similarly, crop waste of others and non-food grasses, in which 400 million could come out of non-food grasses, transgenic crops, 500 million biomass, and rice would actually beat that with just the crop waste, 731 million mg. And that essentially, which we can increase the efficiency of conversion, increase the cellulose by 30%, would be therefore much more useful to than to engineer many other plants, just use the biomass. So that's an alternative which we are working on, whether or not it's taken. I gave a talk like this in Korea once upon a time, and they're interested in doing something. Rice is a waste in many parts of the world. So these are the people who worked on it. Papers by Madan, Arjun, and Batlang have already been published on the AT shine, and present work on the OS shine is being done by Ritika Mihani and Supratim Basu. Chirag Gupta is a bioinformatician. Others been involved in also bioinformatics, and transformation of certain plant difficult rice genotypes by Niranjan. So, well, thank you for your attention.
Any question for Dr. Pereira?